This is a recording for the lecture 20, chapter 14, Principles of Diseases and Epidemiology. There's a lot of vocabulary in this lecture for you to memorize and very little concept. The concepts should be very straightforward and easy to follow. As always, you are responsible for knowing all the vocabulary terms and the concepts and the examples given. So let's start the lecture with terminologies. So pathogens, we've learned a lot of pathogens in class, especially after learning about some of the bacteria, worms that causes disease, and viruses that causes disease. So what is a pathogen? Pathogens are basically disease-causing microorganisms. And pathology is basically the scientific study of disease, which involves a number of things. One of which is the study of the cause of a disease, which is basically etiology. So, etiology is basically the study of the cause of a disease, and it's very important to know which organism or how a disease is caused. So, the CDC, which is the Center for Disease Control, is very involved in understanding the disease process by and trying to learn how to control the disease by understanding its etiology. Pathology also involves pathogenesis, which is the manner in which a disease develops, and it's also very important to understand this process too, because if you understand how disease develops and what causes it, and then you should be able to then find a way to hinder its progression or stop it from spreading. Pathology also involves the structural and functional changes that's caused by the disease and the disease effects on the body itself. An infection is the invasion or colonization of the body by pathogenic microorganisms. This means that the microorganism has either gone through your skin or into your body and penetrating through the tissues and through your immune system, and or it's then starts to attach and then start multiplying, and that's the colonization portion. Now, not all infections will lead to a disease. A disease occurs when the infection results in a change from the healthy state of the body, meaning that you get sick. So anything that's different or any pathological changes that's not physiological. Physiological, that term usually means that's under normal conditions. So physiological means normal. Okay. Now, not all infections will lead to a disease because your body is able to overcome that infection with its immune system. And sometimes you may have an infection without any detectable diseases, meaning that sometimes the disease itself is masked and the organism is multiplying, but your immune system is not able to detect it, and so you won't be able to see the disease take form until maybe sometimes when it's too late. Okay, more terminologies. You guys remember we talked about the normal flora, or otherwise known as the normal microbiota in your body. These are basically microorganisms that have already established itself or is living inside of your body and is really not causing any harm to your body. So no disease and no harm is done under normal conditions. So we're not talking about under conditions where your immune system is compromised. So animals that are born in the laboratory during research, they show that these animals, if it's sort of like under germ-free conditions in the laboratory, they discover that these animals, these mice, are not sick. They don't have bacteria or any germs, and it is believed that animals are generally germ-free in the uterus. Well, actually not, yeah, in utero, meaning in the uterus, prior to being born. And then the animals then acquire the microorganisms when it passes through the female's birth canal, meaning the vagina. And how it acquires the microorganism is basically the fact that the female's vagina, there is a normal flora, and that normal flora is usually composed of lactobacillus, or species of the genus Lactobacillus. And so when the baby goes through the birth canal, it will then acquire some of the organisms on its body, some in its eyes, and of course, some will get into its mouth, swallows it, and makes its way into the intestine. Now, this is actually normal and is believed to be a good thing, and that it's believed that this is actually very important for the babies to acquire these microorganisms naturally via this way versus do cesarean sections. They believe that this is actually boosting the individual's immune system by exposing them to the microorganism this way, and also by introducing a microorganism that should be a part of the normal flora in the baby itself. Another example is E. coli. E. coli is another natural inhabitant of the intestines, the large intestine, and E. coli gets into the large intestine by consumption of food products or basically anything that will contain the E. coli, normally through food products that's eaten. Now let me go back and comment on the animals that are born in the laboratories that are germ-free. These animals, although they don't get sick in the confined sort of area because it's sterile environment, they don't get sick. However, they have an underdeveloped immune system, and so when they get older, when they are exposed to the microorganisms, either bacteria that normally won't do any harm to the normal mice, these mice will get sick and they will easily die because they haven't developed a proper immune system. Now, transient flora or transient microbiota, these are microorganisms that may be present for a short time and then disappear. So these are the organisms that will, basically are on the surfaces of your body or inside the lining of the organs in your body, such as the eyes, the nose, the throat, the mouth, the skin, the large intestine, urinary and genital systems, and they're transient because, well, you wash them off, like, or for the skin, you wear clothes, and each time you take off your clothes, so you rub against something, you remove the microorganism. When you wash your hands, you remove the microorganism. But then once you touch something else, then you introduce a microorganism on those areas again. Same thing with your mouth. You brush your teeth, and you eat. You're actually removing microorganism, and you're also when you're ingesting, then you're reintroducing the microorganism. Large intestine and the other areas were basically the same principle. So microorganisms are not absolutely essential for animals because, again, of studies in mice in the laboratory. But as we mentioned earlier, mice that are born in a germ-free environment, they do have an underdeveloped or undeveloped immune system, and they do get sick and can die from exposure to microorganisms that usually do not cause diseases in the other organisms. Now, there's something known as microbial antagonism, which is also known as competitive exclusion. We kind of talked about this before in class, where this is with regards to bacteria fighting each other for space and for nutrients. Competition among the microbes basically allow the normal flora to prevent the overgrowth of some harmful microbes that had just entered the body and is trying to find a space to then attach to and multiply and to replicate and to multiply in mass numbers. And so having the normal flora is actually a very good thing in this manner. For example, the normal lactobacilli, so from the genus Lactobacillus, the species from the genus Lactobacillus that reside in the vagina, they're able to undergo fermentation and then produce a pH environment that's pretty low, about 3.5 to 4.5. And as you recall, when we talk about the optimal pH for most fungus, it's about lower than a 6.5. It's lower than 6, around 5 or 6. And so if you have a pH that's around a 3.5 to a 4.5, this pH condition is too low for even some fungal growth. And so this will be then ideal to control the growth of Candida albicans. And remember, Candida albicans is a fungus that can cause vaginitis, basically a yeast infection. So therefore, it's possible that if you are too clean and you clean yourself too often, down there for some females, what will happen is that you will remove these organisms, the natural flora, and then you'll make room for potentially other organisms such as Candida to then reside, take over, and potentially then cause you to have a yeast infection. So there is a fine line between being too clean or being too dirty. So everything has to be done in moderation. So now let's look at the various different types of relationship that can be formed between the normal flora and the host. The normal flora and the host forms a symbiotic relationship. So you guys remember what symbiotic means. Sim is together, bio is life, still living together. And so there are three types of symbiosis that can be formed between the bacteria or the normal flora in your body and you, the host. So the first example is commensalism. This type of symbiosis is a form in which one organism benefits and the other is unaffected, meaning there is no harm to the other. So this usually refers to the microorganism that, like, probably lives on your skin. It's not really doing anything except for potentially just in terms of not allowing other microorganisms to grow in its place that can be pathogenic. So many of them, your normal microbiota in the body are commensals. Commensals is a term that's used to refer to then organisms or microbes that form a commensal relationship with the host. Corynebacteria is an example of a microorganism that lives on the surface of the eye, and this we're talking about the non-pathogenic form. Okay. The second type of symbiosis is mutualism. Mutualism, like it says, is a mutual agreement between the organisms. So it's basically a type of symbiosis in which both organisms benefit from the relationship. So one example is E. coli. E. coli lives again mainly in the large intestine of humans and animals, and the E. coli benefits from the host by one, it receives a home, a place for it to inhabit, and two, it then can receive some nutrients that has not been absorbed by the host but has been passed along to the large intestine. And what the host gets out of the E. coli is that the E. coli then is able to synthesize vitamin K and some B vitamins for the host to utilize. And these vitamins are important in forming blood clotting proteins. You guys probably have heard of the term probiotics a lot of times, and especially in commercials now, with, I think, the yogurt Activia. And so some people lack enough E. coli in their GI tract, and so they have problems with digestion. And so what these companies do is that they claim that if you eat certain products such as the Activia yogurt that's sprinkled with the probiotics, the E. coli, or, well, not necessarily E. coli, but I believe this contains lactobacillus. The organism will then to be ingested and they don't die. Well, some of them will die, but some of them will make its way into your small intestine and then into your large intestine and colonize those areas and, of course, help you digest food better. So an example of bacteria that's usually commonly used in these probiotics are what you call labs. The lab LAB stands for lactic acid bacteria. Again, the common one is lactobacillus. The last form or type of symbiotic relationship that is formed between the normal flora and the host is parasitism. Parasitism is a type of symbiosis in which one organism is benefited at the expense of another organism. So, at the expense of another organism means that it's commonly causing some harm to the host. Um, so, you're looking at some viruses and some of the pathogenic microorganisms that you studied so far. And of course, in some cases, in humans, some people can refer to like the babies as parasites because they are living inside of the mommies who are the host and absorbing their nutrients, and it's not really, the mom is not really benefiting from the baby, in some cases having feeling more tired, fatigue, loss of memory sometimes. So one can consider that as a parasite, but of course, don't tell everybody that. And sometimes the parasitism can last until the age of 30 or even 40. Sometimes the relationship can change. So it can go from maybe commensalism or mutualism to something like an opportunistic relationship. So we've discussed about opportunistic pathogens before. These are basically pathogens that are potentially pathogenic organisms that normally do not cause diseases in the individual, but can cause a disease when there is a drama, not drama, but trauma done to the host itself, something that will cause the immune system to be decreased or lowered or affected in some way. Some examples are Neisseria meningitis and E. coli. Neisseria meningitis are organisms that normally resides in the respiratory tract at low numbers. They're fine, they're benign, they don't really cause any problems. However, when conditions change and they multiply in a higher number than normal, or your immune system is compromised to allow them to multiply, and again, higher rate than normal. And then they can cause meningitis. And you can remember that meningitis is basically inflammation of the meninges, which are the coverings, the protective layers that covers the brain and the spinal cord. Okay. E. coli, remember, form a mutualistic relationship with the host in the large intestine. However, when E. coli gets into an area that's not the GI tract, so for example, let's say the urinary tract or in the lungs, it can then cause other problems. So again, if it goes to the urinary tract, it can cause urinary tract infections, and it can also cause meningitis or other serious health problems if it gets if the E. coli resides in areas other than the large intestine. Now let's look at the etiology of infectious diseases. So remember that etiology is basically the study of the cause of a disease. And so how one can study the cause of a disease is by using Robert Koch's postulate. Remember back in lecture 1, when we talked about the scientists and their discoveries, how Robert Koch's postulate was able to link a disease link an organism to a disease. And I would like you to go back to that particular lecture and review your notes on that. Right now, I'm just going to summarize this cautious postulate for you very briefly, but I am going to still hold you responsible for knowing the details of the outline of the experiment. So recall that Pasha's postulate is basically, again, trying to link an organism to a disease, and the experiments is as follow to show this. So you then, the scientists will have to be able to extract the microorganism from the infected host. Let's say in this example, the mouse. It's infected with the organism and it's dead or it's really sick. Then the microorganism is then cultured and examined under the microscope, and other biochemical tests are performed to learn more about the characteristics of the microorganism. Then a sample of the microorganism is injected into a healthy specimen, so another healthy mouse, And then the scientists would observe to see if anything happens to it, and will then take note to see if if the disease occurs, to see if the same pattern of the disease occurs, like in terms of signs and symptoms are similar or not. And so, if the organism is dead or shows the same pattern of disease, then a sample of the microorganism from the blood or tissues of the specimen would then be obtained, studied under the microscope again, or do biochemical tests to see if if the microorganism that's isolated is the same as the original. And if it is, then you can safely say that this particular microorganism that you have extracted and isolated is the cause of this particular disease. And slide 10 is a short summary of what I just mentioned, but it's written out. There are exceptions to cautious postulates. One exception is that some microorganisms or some organisms cannot be grown in pure culture itself. So, for example, Treponema pallidum, which is a causative agent of syphilis, cannot be cultured on artificial media. So, you have to use special techniques to culture them. So sometimes you just aren't able to isolate or identify the organism itself due to our inability to culture the organism. The another exception is that when an organism is a parasite, is especially an intracellular obligatory parasite that needs the human as a host, you can't use human subjects, test subjects. Nowadays you can't because of human rights activism, because it's just morally wrong to do that. But in the past, the people have been used as test subjects. So, for example, King George the First back in about 1721. I'm pretty sure this has been done even before 1721. But what King George the First did was that he used prisoners as test subjects for the test on smallpox vaccine. And so he told the prisoners that if they volunteered as test subjects, if they survive the process, then they get to go home for free. But if they die, then oh well, you die. That's a chance or risk that you're taking, because it could be a better chance than staying in prison back then. Because back then, the prison is you don't get 3 meals a day. You don't get trips to read books from the library or get a degree. Back then, they probably starved you and whipped you to death. Okay, now we're going to switch to classifying infectious diseases. There are several ways to classify infectious diseases. One is through the usage of symptoms, signs, and syndromes. A symptom is basically a change in the body's function, such as pain and malaise. Malaise is just basically not feeling well. And these are just basically very vague feelings of the body in terms of discomfort, but it can't be really measurable. Signs are any changes that can be observed and actually measured. So this is more objective, versus symptoms are subjective. So examples of signs is things such as lesions, swelling, fever, paralysis, things that you can observe, the doctor can observe, other people can observe, and can basically measure and evaluate clearly. Symptoms, things like, oh, I feel pain. Well, there's no instrument to measure pain right now, except for using a survey. So when you go to a doctor, you go, oh, I don't feel well. This hurts here, and then the nurse or the doctor would ask you, well, on the scale of 0 to 10, how much pain do you feel? 10 being the most pain, the worst pain that you can't ever, that you can't tolerate, basically, and 1 being just like things that you can tolerate or nothing. So that's not something that's very concretely measurable. That's how you feel. Same thing with tiredness. How tired are you? I don't know. I mean, one person's tiredness is different from another person's exhaustion. Okay? A syndrome is a specific group of symptoms or signs that always accompanies a particular disease. So when you get into the nursing program, you see that certain diseases has particular signs and symptoms. And so you have to memorize these signs and symptoms. So when you look at a person, you'll be able to classify, well, this person has this disease or maybe this person has that, based on, again, the signs and symptoms that they see. You can classify a disease based on how it behaves within a host. So it can either be communicable disease, contagious disease, or non communicable disease. A communicable disease is any disease that spreads from one host to another directly or indirectly. So examples of communicable diseases are chickenpox, measles, genital herpes, typhoid fever, and tuberculosis. Communicable again means that it spreads easily from one host to another directly or indirectly. Directly means you have to touch someone, and the disease is either on their person or the body, or they sneeze directly on you, and the pathogen is in the aerospray. That's, well, meaning that their spit or coming from their nostrils, mucus. And of course, in the hand, if all those bodily fluid gets on the hand and you touch someone else, and that person then touch their body parts and inhale it or get it into the system. Indirectly is basically passing the pathogen onto, say, a handlebar or on the doorknob, and then another person touches that doorknob and then touches themselves. That is indirect. Contagious disease is a disease that can be easily spread from one person to another. A contagious disease is also a communicable disease, but it's contagious means that it's worse than communicable because it can easily spread from one person to another. So chickenpox and measles are communicable diseases, but they're also contagious diseases. Non-communicable disease is a disease that is not spread from one host to the other. So examples, tetanus. So Clostridium tetani is the organism that causes tetanus, can only cause a disease when introduced into the body via abrasions or wounds, meaning that you have to have some sort of an open cut through your skin or to the other parts of your body, and then it has to be able to then penetrate inside that way. So you won't be able to get tetanus if somebody has tetanus and they spit on you. You're not going to get that unless there is a open wound on your face because somebody used to scratch you, or you just scratch yourself and it's an open wound and then it penetrates that way. So that's non-communicable disease. Classification can also be based on the occurrence of a disease. So there are terms that you're going to see frequently when you get into the health field, especially if you're going to be a nurse. Terms such as incidence of a disease versus prevalence of a disease. Incidence is basically the fraction of a population that contracts it during a particular period, while prevalence is the fraction of the population having the disease at a specified time. Now, what you're thinking, well, don't they sound alike? Yes, they do sound alike, but the difference really in incidence versus prevalence is that prevalence takes into account the number of cases that the individuals have in the populations that have that disease, and it takes into account old cases and new cases, versus incidence just talk about those cases, the new cases within a particular period or time. So, for example, the incidence of AIDS in the US in 2004 was about 40,000. So, again, the incidence of AIDS in 2004 was 40,000. And these are only cases, new cases that occurred in the year 2004. The prevalence of AIDS in 2004 was 900,000. So this is a larger number, 900,000 versus 40,000 for incidence. So prevalence then takes into that 40,000 incidents in 2004 and all those cases before 2004. So like, 1997 or even before then. You can also classify a disease based on its frequency of occurrence. So, sporadic disease is a disease that occur occasionally, such as typhoid fever, so once in a while. Endemic disease is a disease that's constantly present in the population, such as the common cold, meaning that it's always existing, no matter what season it is. Epidemic disease is a disease acquired by many people in a given area in a relatively short period. So, an example would be influenza and AIDS. Now, sometimes an epidemic disease can become a pandemic disease. So, a pandemic, pan meaning cross, a pandemic disease is an epidemic disease that occurs worldwide. So, it's going to cross continents. It's not only going to happen in the US, it's going to happen in Mexico, it's going to happen in Canada, it's going to happen in Europe. So it's worldwide. So, for example, influenza, say the avian influenza, or the HIV that causes AIDS disease. HIV is not a disease. HIV is a virus, but AIDS is a disease that's caused by HIV. So AIDS originally started as an epidemic, and then it became pandemic when it was discovered that people around the world can also or did acquire that disease through the HIV virus. And epidemic disease can easily become a pandemic disease. And actually, this is one of the fear that the CDC has, is that one of the influenza, the avian flu that occurred a while back in China. No, I believe it was in Taiwan. They were worried that it would become pandemic. And the reason why that it would become pandemic easily is because of the easy, transportation that one can get. So one can travel easily from Taiwan to the U.S., to Europe, to Antarctica, anywhere easily via the airplane or other means of transportation like trains and whatnot. But the airplane is the easiest way to transport the disease from one area to the next. I would like to recommend a movie called Contagion. At the end of the semester, once you're done with the class and you have all this knowledge and you're tired and you poop and you're done with your Christmas shopping, rent a movie and watch it, the Contagion movie, and watch it with your friends. And you'll see how much you've learned about microbiology that is in that movie. You see how things can, a particular disease can easily be passed, how it can become communicable to becoming contagious, and then becoming epidemic to pandemic. And listen to what they were saying, that what they talk about the virus itself and how fast it can mutate. I'm going to skip slide 16, which is a graph to represent the reported cases of AIDS in the US, so you don't have to worry about that. One can use severity or duration of a disease to classify it and to define the disease. So, an acute disease is one that develops rapidly but lasts only a short time. So, influenza is an example of an acute disease. In other cases, it may not be an acute, but it can be an acute disease, meaning you develop it really fast and then it goes away fast. The next one is chronic disease. Chronic time means that the disease develops very slowly. It's a less severe reaction at first, but this disease, because it develops slowly, is continual, or it could be recurrent for long periods. So, for example, tuberculosis, syphilis, and leprosy. Between acute versus chronic, usually chronic is a worse because the disease takes a really long time to develop and your body takes a while to react to it. By the time you really feel something, it may be too late. Acute could be better thing in that the reaction is fast, and you're able to react to it really quickly and get the drugs for it to treat it. However, an acute disease can also be bad in that it develops so rapidly that you just aren't able to, your body can't respond as quickly, and so you can die for something that is acute. Subacute disease is an intermediate between acute or chronic. So here we're just talking about duration, time span. So acute disease, we're talking about, depending on the disease, you can talk about something that is, days to weeks to a few months. Chronic can be, depending on the disease, can be months or it could be years. Subacute disease then can be somewhere between acute and then chronic disease. A latent disease is a disease when the causative agent remains inactive for a time, but then becomes active to produce symptoms of the disease. So, for example, shingles. So shingles is caused by the same virus that causes chickenpox. And so when you get chickenpox, it clears and then you think it's gone. But then, so then the virus basically lays latent for a while, dormant. And then, under stress, the virus itself manifests and it becomes shingles. And so that's what it means by it becomes active after a long period of inactivity. Okay? Also classify disease according to its effect on the host's body. A local infection is basically an infection in which the invading microorganisms are limited to a small area of the body, meaning that it's the area where the infection occurs. So if the infection occurs there, you'll see the organism having its effect right in that area. A systemic or generalized infection, systemic, you're thinking about the whole system. This is when the microorganism, or sometimes not the microorganism itself that's causing a problem, but the toxin that it releases that causes a problem, are then spread out throughout the body via the blood vessels or the lymphatic vessels. So because the blood vessels and the lymphatic vessels are pretty much the freeways and highways of your body, and so once the microorganism get into your vessels, they can travel everywhere and then exit and deposit themselves in other regions of the body. So example of a systemic infection is measles. A focal infection is it's basically an infection which agents of a local infection spread to other parts of the body and then become confined to that area, meaning that the organism gets in and it doesn't causes its problem there. That would be a local infection. What happens is it goes in, it has an infection there that's local, and then it starts to migrate and goes elsewhere to cause then another infection at another area. So that would be then a focal infection. The second part would be then the focal infection. Bacteremia, emia is basically blood, and so bacteremia is basically infection of your blood in which the microorganism is bacteria. Septicemia is basically blood poisoning, and it's usually systemic infection because you have the microorganism in your circulatory system, so systemic infection, and this is due to the bacteria multiplying in your blood. Toxemia is basically then is like think of toxin. Emia, you're thinking of blood. So toxemia is basically you're detecting signs of the toxin of the microorganism that's released into your blood. And this could be just as bad and as deadly as septicemia. Viremia then is basically detection of viruses in your blood. And therefore then this would be considered to be systemic infection because it is in your blood vessels. A primary infection is basically an infection that is an acute infection that causes the initial illness. And then sometimes you can get a secondary infection, which is basically is caused by an opportunistic microbe after the primary infection has weakened the body's defense. Basically, what they're saying is that first you have a primary infection, and the primary infection causes you to be really sick, decreases your immune system or lowers your immune system. So, one really good example is HIV. The HIV virus infects you first, attacks your T cells, which are important in your immune system. So once your T cells are gone or lowered, then your body is unable to fight off any additional infections. When you have an additional infection, such as in the case of HIV patients, it's usually some respiratory infections that can cause pneumocystis pneumonia, and this is usually what causes the individual to die. So again, the primary infection from the HIV can give you AIDS, but the individuals won't die of AIDS, but they'll die of secondary infections, usually some complications with the respiratory system. In inapparent or subclinical infection, so the key term is inapparent and subclinical. Inapparent means you can't see it. These infections do not cause any noticeable illness, so you don't really notice that you're sick physically until sometimes later. And this is usually sometimes something that's referred to as a chronic disease. So, for example, hepatitis A and polio, they may be carried by people who never developed the disease, but they're infectious. Or they can also be just that they don't develop the disease, or the disease develops really slowly. Okay, and this is kind of scary because sometimes individuals who have inapparent infections, because they don't really know that they're sick or that they're carrying the infection, they are the ones that can easily then spread the infections to other individuals because they don't know, so therefore they don't take extra precautions. All right, now let's switch to the spread of infections, how infections can be spread. So, for disease to perpetuate, mean to continue on, there must be or there has to be some sort of continuous source of the disease organism. So that means that they have to be a reservoir of the organism somewhere. So, reservoirs of infections are basically sources of living organisms or inanimate objects that provide pathogens and environment for survival, multiplication, and transmission. And so reservoirs can either be human, animal, or non living objects. So let's look at human reservoirs. These are basically people with signs and symptoms of the disease who then may transmit the disease to other individuals. Carriers are usually referred to as individuals who transmit the disease, but they do not exhibit any signs or of illnesses. So we're looking at, so these people, the carriers, have what you call an inapparent or subclinical infection. So remember, I mentioned that these are the individuals who are the scary ones because you don't know they have it, they don't know that they have it, and they can easily spread it to others. The second type of reservoir that we're going to look at are animal reservoirs. And this is usually zoonoses, and zoo is basically animals. So zoonoses are diseases that occur primarily in wild and domestic animals, and then they can be transmitted to humans. So these animal reservoirs are like, say, bats, skunks, foxes, or dogs. These animals usually carry some sort of organisms that can easily infect others. So, well, not infect others, but infect humans. So rabies is an example of a disease that can be transmitted by these animals. There are about 150 zoonoses that are known. And remember, when we talk about zoonoses, we are talking about diseases that occur in wild and domestic animals. Okay, non-living reservoirs are inanimate things. Examples of non-living reservoirs are soil and water, and these are actually the two major non-living reservoirs. Water, because it contains a lot of fecal matter, and soil, well, you know that there are a lot of microorganisms, lots of bacteria and fungus that live in soil, in addition to worms. Remember, we talked about that in the previous lectures. Now let's discuss about the three major routes of transmission of disease. The first route is via contact transmission. This is basically the spread of the microorganism by direct contact or indirect contact, or via droplet transmission. Direct contact, like I mentioned earlier, is that you have to physically come into contact with the source and the host, meaning you have to touch the host somehow, and that person is passing it from one person to the other. Indirect contact is basically means that the microorganism is being transmitted from the reservoir, one of the reservoirs that we mentioned earlier, to a host via a non-living object. Usually the non-living object is referred to as a fomite. So that's a definition of a fomite, actually, or non-living objects that are reservoirs that contain the microorganism that can be spread to other individuals. So real easy examples of fomites are tissues, especially when you have the cold or the flu. The organisms are in the tissues that the infected individuals has sneezed into. So the bodily fluids are in there. Diapers, and because of poop. Syringes, again, the needle goes into the body, blood. So there's blood, and if the organism are in the blood, then you can have it on the syringe itself, and then if somebody else injects it into their body, then you're transferring the microorganism into their body that way. Droplet transmission is referred to as basically discharge that you have put into the air. So you're sneezing or coughing or laughing and talking, and you spit, or you spit your saliva, and your mucus has traveled into the air. Okay, and the key is that these droplets they travel less than 1 meter, because it's beyond 1 meter, you call it something else. Okay. So slide 23 shows a series of images to give examples of the different types of contact transmissions. So direct, so you can see here direct in the left panel with the lots of individuals, people touching each other at a party. Actually, at parties, those are the times when you have easy transmissions of microorganism, directly or indirectly. And then, of course, the bottom panel, lower right, shows an individual sneezing, and you have this fast camera that's able to capture the droplets in the air when you sneeze without covering yourself. So when you get sick and you're sneezing or you're coughing, the best thing to do is to cover your mouth and your nose, so that you do not spray the microbes around. And best thing to do is to not cough into your hand, because then your hand touches other things, and then you're transferring it into fomites that can then be picked up by other individuals. So the new rule of thumb is that if you need to sneeze or cough into your armpit area, okay, to prevent it from spreading. And of course, in the future, when you become health caretakers like nurses or surgical nurses, and of course, doctors taking care of patients, you need to then be able to prevent these form of transmissions, well, prevent the microorganism from being transmitted via one of these forms by taking precautionary measures, by wearing like scrub uniform, I'm wearing a mask, and of course gloves. It's very important to wear gloves. The next major route of transmission is vehicle transmission. This form of transmission this form of transmission is you're basically transferring the microorganism by a medium, and this medium could either be water, food, air, blood, bodily fluids, or intravenous fluids. Bodily fluids can mean like urine. So let's look at waterborne transmission, which is a type of vehicle transmission. Waterborne transmission, you're basically spreading the pathogen due to the water that's contaminated by untreated or poorly treated sewage. So meaning fecal matter has gone in. So an example would be cholera, shigellosis, and leptospirosis. Foodborne transmission is a transmission in which the pathogen is transmitted in foods that are incompletely cooked, poorly refrigerated, or prepared under unsanitary conditions, meaning that the individual did not wash their hands properly. And remember, we talked about how important it is to properly cook pork, especially if there are cysts of worms inside of the meat, of the muscles. And of course, if it's the food is poorly refrigerated, then the microorganisms such as bacteria can grow at a faster rate than if it has been refrigerated, which would then decrease the metabolism and is have more of a bacteriostatic effect on the bacteria. Airborne transmission is basically the pathogen is transmitted in the dust that travels usually more than 1 meter from the reservoir to the host. So remember, droplet transmission is that the discharge is traveling less than 1 meter, but airborne is more than 1 meter. So here we're talking about, like sometimes like those people, gardeners with their packs, and they're blowing the dust all over by cleaning somebody's yard, and you're walking by, and so you're inhaling all this dust. And remember, inside the dust of soil, there are spores and pathogens traveling within. I think somewhere in Santa Monica, they're trying to ban these blowers, leaf blowing kind of equipment. I also want to touch upon cholera. This is usually prevalent in areas where the sewage condition is not treated properly. So, especially in the case in Hurricane Katrina, that caused problems, and so people don't have the proper usage of toiletries, and so there are cases of cholera broken out there, and not so much as in, say, Haiti. I think that was the area a couple of years ago or last year. Lots of cases of cholera breaking out in that region too. Slide 25 basically shows the different types of vehicle transmission: waterborne, airborne transmission, and of course foodborne transmission. Lots of food poisoning cases can occur with foodborne transmission from individuals that work in the kitchen and do not prepare the food properly, or unsanitarily not wash their hands. And of course, the waterborne one, they're showing an individual drinking water from a stream. So, mountain spring water. Sounds great. It's refreshing. It's cold. It's cool. It tastes great. But along with that nice taste, you might be ingesting fecal matter from an animal from the wild that has done its thing, maybe upstream from where you just drank your water. And you know that there are microorganisms in feces and in the urine of all animals. The third major route of transmission of diseases is vectors. Animals, basically, vectors are animals that carry pathogens from one host to another. The common vectors are the arthropods, and arthropods are basically animals that have jointed appendages. So insects are common arthropods that you know of. Examples are like flies, ticks, fleas. Those are arthropods that are common vectors. So with vectors, we have different examples here. We have mechanical transmission and we have biological transmission with vectors. So mechanical transmission is the transport of microbes on the insect's feet or body parts. And you guys know the flies are everywhere. They go everywhere, and they like to visit dumpsters because flies are attracted to decaying matter. They like to lay their larvae in the matter, and the larvae then basically live off of the nutrients from that substance. So then basically then when the fly lands on either poop or trash with microorganisms, their pads are sticky, the ends of their feet are sticky, and so I shouldn't say feet, I should say appendages are sticky, and so the microorganisms are attached to those appendages, and then they would then fly to you and land on your food. And when they do land on your food, then the pathogen that's on their appendages will then be then transferred to your food, and then you would eat that food, and then you would ingest those pathogens or microorganism into your body. And then if your body is unable to clear the pathogens or defend it or eat the toxins from the pathogens, then you get sick. So examples are typhoid fever and bacillary dysenteries, or insects, microorganism that causes these diseases can be transferred via mechanical transmission from these vectors. See, biological transmission are basically means that the arthropod, the vector, has to then bite an infected individual and then transfer the microorganism directly into the person's body with the bite. So, for example, is the mosquito, the Anopheles mosquito, and Plasmodium that causes malaria. And so, when the mosquito bites the human, it then transfers its saliva, and saliva has the if the saliva has the infected sporozoite form, it will then enter into the human's bloodstream, and then that's how the human will then get infected. Usually, the microbes that infect the arthropods require the arthropod as one part of its life cycle. So remember, in the case of Plasmodium, the gametes of the Plasmodium that's in the mosquito's intestine or stomach, it's then transformed. The gametes then transformed, and they unite to form the zygote, and then they divide and produce more sporozoites. So the arthropod is an important stage of this life cycle. Okay, now let's talk about nosocomial infections. Nosocomial infections are basically hospital-acquired infections. About 5 to 15% of our hospital patients acquire some form or type of nosocomial infections. So, you go into the hospital, you may come in with one type of an infection or disease or illness, and then you might come out or with another form of infection, and that infection may come from the being in the hospital itself. In the United States, approximately about 2 million people per year contract nosocomial infections. And of those 2 million people, about 20,000 of them die as a result of the nosocomial infection. So that's a bit too many for my liking, and probably for you too, knowing that there is a chance that you may die when you walk into the hospital. Um, it is nosocomial infection is currently known as the 8th leading cause of death. And nosocomial infections usually result from the interaction of three things. So these three things, three factors, have to come into play, and they have to intersect in order for a nosocomial infections to occur. The first condition is that the microorganism has to be present in the hospital in order for you to acquire the infection in the hospital. The second factor is that the individual, the host itself, has to have some form of a compromised or weakened immune system. And the third factor that needs to come into play is the chain of transmission in the hospital. And we'll talk about each one of these factors in a few minutes. Let's look at, a few seconds actually. So let's look at the microorganisms that are involved in the nosocomial infection. So they have to be present in the hospital for you to get these. Okay. So most of the microbes that cause nosocomial infections do not normally cause a disease in healthy people. These are usually not pathogenic, and they're only pathogenic in the weakened individuals with a weakened immune system. So we're talking more about opportunistic pathogens, and again, you know what those are. So here are examples of opportunistic pathogens in the hospital. I do need you to know the categories and be able to give me examples. Okay. So enterobacteria. So these are organisms that causes problems usually in your GI, but not exclusive. So enterobacteria includes E. coli, Klebsiella, or species within the genus Klebsiella, species within the genus Proteus, species within the genus Enterobacter, and Serratia marcescens. These organisms account for about over 40% of the nosocomial infections in the hospital. These can cause infections such as urinary tract infections, peritonitis, meaning inflammation of your peritoneum, bacteremia, pneumonia, septicemia, and gastrointestinal inflammations, neonatal meningitis. These are just some examples. It's not exclusive, nor are these only the ones that are known. Staphylococcus aureus, we've talked about this organism, this microbe, in some extent in the previous lectures. We work with it in lab. So about 16% of nosocomial infections are caused by Staphylococcus aureus, and it can cause urinary tract infections, respiratory tract infections, endocarditis, which is basically inflammation of your endocardium, which is the lining of your heart, innermost lining of the heart. And the scary thing about Staphylococcus aureus is that it is rapidly becoming highly resistant to antibiotics. Meaning that common antibiotics are not able to be used to treat Staphylococcus aureus. So you're going to have to use more potent forms, and the more potent the drug that is used, the higher the side effect risk. The other group that we're going to look at are Enterococcus. Enterococcus account for about 10% of the nosocomial infections, and then species within the genus Enterococcus can cause urinary tract infections, surgical wound infections, and of course also endocarditis. Or it can also be pronounced as endocarditis. Pseudomonas aeruginosa, you're also very familiar with this bacterium. It accounts for only about 9% of nosocomial infections. It can cause infections such as burns and surgical wound infections, septicemia, pneumonia, and these again are organisms that are highly resistant to not only disinfectants but antibiotics. Again, because they have an R factor, which carries genes that allows it to be highly resistant to antibiotics, and because of the small porins and pumps. The second factor that causes nosocomial infections is a compromised host. Meaning that is a compromised host is basically a patient or individual whose resistance to infection is impaired, either by burns, because the burn has destroyed the skin, and the skin is the first mechanism of defense in your body, or that the patient itself has an impaired immune system due to disease or therapy. So two conditions that can compromise the host are the following. Is again, the first one is broken skin or mucous membrane, and again we mentioned before that the skin and the mucous membranes are basically protection against invasion. And so when you have burns or surgical wounds or trauma or injection that pierces through the skin, or any of these things, you can basically then can easily introduce the microorganism into the body. The second condition is that you have a suppressed immune system, and meaning that there is something wrong with your T cells or your B cells most likely. This can be a result either to not only caused by a microorganism or virus, but it can be caused or due to taking drugs that can affect these cells or the cell division of these cells. Radiation therapy, and again, remember with radiation, you can affect not only cells of your immune system, but other cells in your body, especially cells that have, but are still able to divide and are dividing continuously, like cells of your small intestine and your hair cells. Those cells that are continuously dividing, they're usually affected by radiation or by drugs. Taking steroid therapy, diabetes, people who are diabetic, leukemia, stress, malnutrition can cause your immune system to be compromised. So any of these things. Okay. The third condition that can cause one to have a nosocomial infection is the chain of transmission. So basically meaning that the routes of transmission from the people working there in the hospital, the staff to the doctor, and how it gets from those individuals or even the objects to the patient. So these are the main routes that usually cause nosocomial infections. One is direct contact transmission from the hospital staff to the patient. So it can either be the staff individual is not wearing protective gloves or mask, or not washing their hands frequently from going from one patient to another, or any reasons. Direct contact transmission from patient to patient. So sometimes the hospital have too many patients and not enough beds, or there's just way too many patients in close proximity to each other. They can then pass the the disease, with basically the organism from one individual to the other. Third is route is indirect contact transmission from fomites, such as catheters, syringes, or respiratory aids, especially if they're not cleaned properly, disinfected properly, or reusing syringes, which shouldn't happen in a hospital these days. The fourth route is indirect contact transmission through the hospital ventilation system, and can be considered to be an airborne form of transmission. So all hospitals and any building these days will have a ventilation system, and so they should frequently clean out the vents and the filters, the air filters in the vent. But if hospitals are cutting back, and if they don't clean their vents regularly or change the filters regularly, then you can have a problem. Okay, so control. How do you then control nosocomial infections? Then the first thing is always using aseptic techniques to reduce the number of the pathogens from patients that the patients are exposed to. So the number one way to reduce this number of pathogens is to wash your hands. So on average, healthcare takers healthcare workers wash their hands approximately 40% of the time prior to interacting with the patients. So this is not very good. I mean, you're not looking at 50%, looking at 40%. So remember that podcast, the science podcast that I had played for you guys a few days ago. They were taking a survey with patients in terms of how comfortable they were with asking their healthcare takers, the nurses and the doctors, to wash their hands. And so it's important that if you feel uncomfortable, or if you want to make sure that your nurse, the person examining you, or the doctors examining you, are using aseptic techniques, you can ask them to wash their hands prior to touching certain parts of your body. Changing of gloves is also important, and usage of isolation wards, or using proper disinfection techniques. Okay. The second way to control it is hospital infections control personnel should ensure that there is proper storage and handling of equipment and supplies. So they're using aseptic techniques and sterile techniques to make sure that they don't contaminate the equipment or supplies, especially with the catheters or needles. Okay, so with slide 37, you'll see an image, a diagram showing 3 circles, 3 areas. And so those 3 areas is to basically indicate the 3 conditions that must be present at the same time for an individual to acquire a nosocomial infection. So I'm going to repeat and say that in order for you to acquire a nosocomial infection, you have to be, 1, in the hospital and acquire the organism in the hospital. 2, you must have some form of a compromised host, so your immune system has to be compromised in some way. And that the third condition is that somehow there is something wrong with the chain of transmission, where either the staff workers or the doctors and whatnot, or the equipment is unsanitary and pass the pathogen to the individual whose immune system is weakened. Okay, so you gotta have all those 3 conditions present in order for the nosocomial infections to occur. Okay, now let's talk about patterns of disease. So the occurrence of the disease depends upon the resistance of the host to the activities of the pathogen. So this basically means that what is the probability or the chances of you getting disease once you have an infection is dependent upon your immune system, or how active your immune system is. And there are some predisposing factors for this. So these are factors that makes the body more susceptible to a disease and may alter the course of the disease itself. So, for example, some predisposing factor is gender. So, females have a higher incidence of urinary tract infection than in males. And this is because the female's urethra is closer to the anal opening or anal orifice than the males, and also the urethra of the females is much shorter than that of the males. And the males is longer because it's basically the length of the penis itself. Okay. Males have a higher incidence of pneumonia and meningitis than females, on the other hand. Other predisposing factors are genetic backgrounds. So, for example, if you have a genetic disorder in which your hemoglobin gene is affected, then you may have the risk of having sickle cell anemia. Other conditions are climate and the weather. Winter brings more respiratory diseases than other climates or weather. Inadequate nutrition, which can compromise your immune system. Fatigue, when you're more tired, your immune system is a bit lowered. There's just harder to fight off infection. That could be one of the reasons why during finals week you have more individuals getting sick than during other times of the semester. Age, the older you are, the weaker your immune system, because basically everything slows down. The production of your cells of your immune system is also lowered. And when you're younger, you're also more predisposed to getting sick because, again, your immune system is not well developed. The environment could be a factor too. The habits that you have, like poor eating habits, unsanitary habits, or habits in which you don't shower frequently, that could be a problem. Lifestyle, in terms of, again, what you eat and when you sleep, or the cleanliness of your house. The occupation could be a problem. Some jobs predisposes you to getting higher infections or more infections than other jobs, such as again, working in the hospital. You'll be more predisposed to getting an infection because you are more exposed to patients who are sick. Or let's say, for example, individuals who work in carpentry or in the housing industry, they may get sick from breathing in certain materials that can cause them to have cancer or other illnesses. Now let's switch to talk about the development of a disease in terms of the sequence of events that leads to the development of the disease. All diseases need to go through a sequence. The particular sequence starts off with the incubation period, the prodromal period, a period of illness that's followed by a period of decline, which is then followed by a period of convalescence. The incubation period is basically the time interval between the actual infection and the first appearance of any signs or symptoms. And remember, we talked about what signs and symptoms are. If you're not clear on that, make sure you go back to the first few slides of this lecture. We call that symptoms is more subjective versus signs is more objective. So the time of incubation depends on the organism itself, the virulence of the microorganism, the number of microorganism that is present, and also the resistance of the host. And all these are important because, if there are small numbers of microbes that infects you, the body can clear that up much faster and take care of it much faster than, say, you have a larger dose of organism infecting you at one point. And of course, if you have a poor immune system, you're not going to be able to fight off the infection. So that could be a problem. So you have a longer incubation period. versus if your body has a good infection, a good immune system, and it will then take care and fight off the microorganism. The incubation period time again varies again depending on these particular factors on you and the microorganism itself. And during this time, during the incubation time, the host, you, actually the individual who is infected may be contagious. Okay, the prodromal period is pretty much characterized by early or mild symptoms of the disease, and this is when you can feel some aches and malaise, which is feelings of discomfort. Then the prodromal period is then followed by a period of illness. And the period of illness is a time in which disease is most severe. Okay, so incubation period, that's when the organism just gets in and it's multiplying. Okay, it's trying to overcome your immune system. The period of illness again is when the disease is very apparent and is most severe. The patients will exhibit very overt signs and symptoms of the disease. Potentially, the number of white blood cells may increase or it may decrease depending on how your body reacts and what the organism or the microorganism that is infecting you. Because the white blood cell can initially be high because your body's white blood cells, which are your cells of the immune system, which includes your lymphocytes and your macrophages, your neutrophils, those initially will be high because they are multiplying and they're trying to fight off the infection. However, the number can decrease if the microorganism are intracellular parasites and they're destroying your white blood cells, like say the HIV or any of the bacteria that we mentioned before that can live in macrophages and destroy them, and so those numbers can drop. The period of illness, within this time, the individual may die if they are unable to fight off the infection. And so, again, this is a very critical stage in the development of the disease, and the individual is definitely very contagious during this period. Then, after the period of illness, it will be the disease will follow a period of decline, and this is when the signs and symptoms decline because your immune system is successfully fighting off the infection, and because your body's working up a lot of energy, expending a lot of energy to fight off the infection, you may become very tired, you may become very vulnerable, and also if some of your organs are compromised, you also then can be vulnerable to secondary infections. For the example of that could be AIDS with HIV. Okay. So then after the period of decline, you can then enter, the disease will then enter a period of convalescence. This is basically when the patient regains his or her strength, and the body then gradually returns to the pre-disease state. So under physiological condition, you may or may not be contagious at this time depending on the microorganism itself. Slide 44 shows a graph that depicts the sequence of events in the development of the disease. On the x-axis is the time, and the y-axis will be the number of the microorganism. As you can see, during the incubation period, the number of microorganisms is low, and during this period, you're going to have the organism will start to replicate and multiply, and your body is trying to fight back the infection. During the prodromal period, that's when you will then show some signs or symptoms of the disease that's developing. And as you can see from the graph, that the number of organism is increasing. And then the period of illness is when you have the most severe signs and symptoms will be shown, and you have the organism is multiplying, is taking over. Then the disease goes through a period of decline, and that's when your immune system is kicking in a high gear and is able to fight off the infection. The number of microorganisms then declines because your cells of your immune systems are killing them. And then you then go through the period of convalescence, and this is when you are recovering and you're getting better, and that the number of organism has decreased, hopefully to zero. But in some cases, you might have residual numbers of microorganism still inside your body. Let's now switch to epidemiology very quickly and very briefly. Epidemiology is just basically the study that deals with when and where the disease occurs and how the disease is transmitted in the human population. The study of epidemiology basically began in 1848 to about 1849, and this is thanks to John Snow, who is a British physician, not the actual Jon Snow in Game of the Thrones. And so basically what happened is that he was a physician, and he studied basically how cholera was transmitted by contaminated water in London during that time. And the key thing about his work is that he was very observant, and he was able to use analytical skills to pinpoint the cause of the problem. So what he did notice was that the individuals in the town who drank water from a particular well got really sick with cholera, versus individuals from the same town who didn't drink from the well. They didn't get sick, but what they were doing is that they were drinking beer, a lot of beer or alcoholic beverages instead of water from the well, and so they didn't get sick. And so then he was able to trace back and figured out that the organism that is probably causing cholera, or actually the source, is coming from the well. And so when he then barred individuals from drinking water from the well itself, the number and the incidence of cholera declined. Epidemiology was also born due to Ignaz Semmelweis, and you guys remember who he was. He was the doctor in the Vienna General Hospital who basically told his interns to wash their hands with chlorinated lime water, because he noticed that the patients in his ward had died because they were passing on the pathogens from the cadavers to the patients, and the mortality rate was about 13 to 18%. And so when he started having his students wash their hands and using aseptic techniques, the mortality rate declined significantly. Okay. So epidemiology is basically the major concern of state and federal public health departments, and because it's very, very important to know when and where the disease occurs, so you can trace it back. And once you know the information there, you can stop it from being transmitted. And the Centers for Disease Control, the CDC, the main center is located in Atlanta, Georgia, and it is the central source of epidemiological information in the U.S. There's a site for the CDC that's free. I think it's called cdc.org, that we can find a lot of information on, and it's free, it's public. And I heard someone told me that the CDC has backup plans in case there is a zombie attack. I'm not sure how, real that is because I haven't been able to find it on the website, but somebody tells me that they have a backup plan in case the zombies attack us. That's kind of scary. In any case, slides 46, 47, and 48, those are charts. Do not memorize everything on that chart. So I'm going to repeat, do not memorize everything on that chart. Normally, I would ask my students to give me 2 examples from each category of selected zoonoses. However, because of time, I'm just going to ask you guys to give me 1 example from each category, but you have to know the following from each example that you give me. So you have to know the disease, the causative agent, the reservoir, the method of transmission. Okay, you don't need to know the chapter for each example that you give me. So what am I talking about? You need to give me 1 example of a viral disease. You need to give me, well, actually 2 examples for bacterial disease, and 1 example for fungal disease, 1 example for the protozoan disease, and 1 example of a helminthic disease. So, so for example, if I ask you to give me an example of a viral disease, its causative agent and reservoir and method of transmission, this is what you need to give me, if for 1 example. So let's say if you pick, say, influenza, then you would say, well, the disease is called influenza. The causative agent is influenza virus. Reservoir is going to be the swine or birds, meaning that the virus itself resides in the swine and birds. And that the method of transmission is direct contact. So you have to come into, the human have to come into direct contact with the infected swine or birds. Okay? So again, 1 example for viral infection, 2 examples for bacterial infections, 1 each for fungal, protozoan, and helminthic disease. Okay. And that concludes the lecture for chapter 14 on principle of diseases and epidemiology. If you have any questions, you can email me or come to office hours.